And new episode of Heredity. What are the learning outcomes of this lesson? So what does it mean that at the end of this lesson what we are going to learn? So this is basically an introduction of heredity. So we will learn the definition of heredity, what is variation, and some other terms which are very important related to heredity. We will learn about Mendel also, but in this topic we will learn today just a brief introduction of about Mendel, and the details of all his experiments we will learn in the coming videos. So let us start with today's episode. So as it is related to heredity, so first we should know what is heredity. So what is it? Now we all know that the young ones always resembles their parents. So that is a well-known dogma of the heredity. So what is it? Heredity is causing similarities between individuals of species. So what is it actually doing? So what is the definition of heredity? So transmission of genetically based characteristics from parents to the successive generation is called heredity or inheritance. Now let us take an example. See, some things you are inheriting from your grandfather or grandmother to your father or either to your mother, and then it comes to you. So it is coming in successive generations. So that is called heredity. So might be it is any of your characters, might be any of your visible features like your eyes, your nose, your hair color, your eye color. So these are all transmission of genetically based characteristics from the parents to successive generation is called heredity or inheritance. Now next comes variations. So by the term variation we actually mean that when a thing or a substance varies from one another. But in heredity, how we are going to describe this term variation? Now once we can say that though offsprings resemble their parents, they are not identical. If you are having brothers or sisters, are they fully identical to you? No. They usually differ among themselves and also from their parents. Even twins, they also resemble each other, but they yet show differences. So differences among the individuals of a species is called variation. So what is it? It is the different genetic characteristics or traits produced in individuals of the same species. Dear students, whatever definitions I am showing here, these are very important for genetics. Please do try to learn them and put it in your mind that whenever in your future you are studying genetics or heredity, you will go through these important terms in every step. So make it clear in the basic step only. So we are clear with heredity. Now the second point was variations. We are clear with that also. So that is the different genetic characteristics or traits produced in individuals of the same species. Now next comes the chromosomes. What are those? If you have gone through my previous slide, you already know the structure and the function of chromosomes. If you have not gone yet through that slide, please go through it. I am putting the link above chromosomes, the video of chromosome, so that you will get a very clear idea of chromosomes. So what are chromosomes? Chromosomes are filamentous bodies present in the nucleus of a cell, composed of chromatin material, DNA protein complex. So a chromosome is a DNA and the protein is the histone. So DNA protein complex. So when you go to learn that chromosome lesson, you will get the detailing of this chromosome structure and function. So it is a filamentous body present in the nucleus of a cell and composed of chromatin material. Now next comes gene. What is it? It is actually the basic unit of inheritance. It is a segment of DNA which is found on the chromosome which controls the expression of a character. So how the character will look like, that expression rather is carried by the gene. and it is passed on from parents to offsprings. That is, from generation to generation, the genes carry that character, expression of that character, and it is a segment of DNA found on a chromosome. So is it clear? What is it? It is a segment of DNA found on a chromosome. It is controlling the expression of a character, and it is passed on from parents to the offsprings. So is it clear to everyone? Please go through it once again, so that you can get it very clear. Let us move on to the next slide. Here comes alleles. So what are these alleles? Now if you have gone through the previous videos, I am again telling you have gone through these terms, but we have not described it fully. We have described in that chromosome lesson also. Now here in this heredity lesson, you will another term you will get, that is homologous chromosomes, which I have already discussed in the previous slide of chromosomes, so I have left it here. So what is it? It is a pair of corresponding chromosomes of the same size and shape, one from each parent, that is one coming from the father and another coming from mother. So that I have put in the cell divisions also, I have told you. So please dear students, go through those videos to get a clear picture of those chromosomes, and cell division is required when you are studying heredity. So in that sequence only I am teaching. Now let us start with alleles here. So what are alleles? Alleles are alternating molecular forms of a gene or a pair of matching genes occupying same position of a chromosome, affecting the same characteristic but in two different ways. So here you can see these two are alleles. So one is dominant allele, which is written in capital letter or represented in capital letter or block letter, and another is represented in a small letter. So why does this vary, one in capital and another one in small? So let us see what does this capital and what does this small signify. So we have told that these are molecular forms of gene or a pair of matching genes occupying same position of a chromosome, affecting the same characteristic but in two different ways. So what is a dominant allele? A super ruling allele that masks any phenotype effect of a recessive allele paired with it. So this is a dominant allele. This big B is a dominant allele. It is masking the effect, phenotypic effect, that is the visible effect, phenotypic effect of a recessive allele. So let us assume that a person is very fat. So that is his dominant allele, and might be either of his parents were thin. So this small b is the recessive allele. So this recessive allele, what is it? It is a heterozygous condition of an allele which cannot express fully or partially in presence of another allele. So as this is a dominant allele, though the mother was or a father was a thin person, it couldn't express itself in that generation, but though it is present there. So this dominant allele will be masking the phenotypic effect of the recessive allele. So the person will be fat. So if the mother is fat, so that is the dominant allele, and if the father is thin, so that thin allele is not expressed fully. It is partially in present, but though it is not expressing fully. Now let us see some examples. So such as freckles, like cleft chin we have seen, and dimples are all examples of dominant trait. You can see that if a father or a mother is having a cleft chin, freckles or dimples, or having almond shaped eyes is a dominant trait, whereas having round eyes is a feature controlled by recessive alleles. And the trait is detached, like detached earlobes, as opposed to the attached earlobes, is also a dominant allele. So there are several dominant and recessive characters which in coming forward we will be studying. So please go through it and make this dominant and recessive allele very clear, so that when we are coming to the experiments of Mendel, in each and every step we will require this alleles. So let us move on to the next one. Now here we will start with homozygous condition. So what is it? Homozygous condition or homozygous chromosomes. Now here we can see the diagrams. So the condition in which a pair of homologous chromosomes carries identical alleles. These two are identical alleles of a gene for a particular character, and here also they are carrying identical alleles of a gene for a particular character. Now this is happening in case of homozygous condition. What is happening in case of heterozygous condition? Then a condition in which the pair of chromosomes carries non-identical or dissimilar alleles for a particular character. In which a pair of chromosomes carries non-identical alleles or a particular character, that is, one is dominant and another one is recessive. Here also one is recessive and another one is dominant. So this is the heterozygous condition in which the chromosomes carry non-identical alleles, and here they are carrying the homozygous or identical alleles. So this is a homozygous condition of a chromosome, and this is the heterozygous condition of a chromosome. So this heterozygous and homozygous I have already discussed also in my previous videos, where I have discussed about the chromosomes. So there also you can go through these two conditions. Now what is there in the next one? Now let us start with a bunch of another definitions, that is character. What is it? Any heritable feature is called a character. Heritable means which is passing on from generation to generation, which can be inherited. That is, heritable feature is called a character. So what are traits? The alternative forms of a character are called traits. What is a genotype? The genetic constitution of an organism is called a genotype. And what is a phenotype? Phenotype is what is externally visible expression of a gene, which is inherited feature is called its individual appearance. So how do the earlobes look like? Does it have a dimple on the face? So that is a phenotype. So these are the examples of phenotypic characters. So are the earlobes attached or they are free? So these are all phenotype. But genotype is the genetic constitution of an organism. We can't actually see this constitution. And what is mutation? Mutation is a sudden change in one or more genes or in the number and the structure of the chromosome in the progeny that had not existed in the parents. So something which has not existed in the parents, suddenly there is a change in the structure of the chromosome that we see in the offspring. That is, mutation has occurred in the chromosomes and which has not existed in the progeny, that is existing in the offspring. Nowadays you can see that many younger kids, they are like CEO of some industry, CEO of certain organizations. Their IQ level is so high. Their parents were not so intelligent and neither did they have so much IQ. So this is due to the mutation of the genes. So that is called gene mutation. So is it clear, all the important terms and topics? Is it clear to you all? If it is not clear, go through it again and again. Whatever textbook you are following, please go through it again and again and learn these all definitions and make it clear in your mind, so that when you are moving forward with genetics, it will be very clear. So now we will go to Mendel's history, a little bit introduction. Now in this slide, let us discuss about who is the father of genetics. Now we have been using the principles of inheritance for thousands and thousands of years. But as a field of science, genetics did not begin until 1866. The year when Mendel published the conclusions of his famous experiments. Now who is this Gregor Mendel? He was born in 1822 in a peasant family, and he then became a monk in an Augustinian monastery in Austria. He had a great interest in plant breeding and hybridization experiments of different plant varieties. Now, breeding and hybridization experiments were his main fields. Over 8 years, he performed hybridization experiments on garden pea plant or Pisum sativum. He was the first to introduce the concept of genes as the basic unit of heredity, and Mendel called genes as factors. Now, he was the first. Gregor Johann Mendel was the first to systematically study, explain the mechanism of transmission of characteristics from the parents to the offsprings, generation after generation. Mendel published his work in the annual proceedings of the Natural History Society of Brünn. Mendel is considered as the father of modern genetics. He was the first to introduce the concept of genes, and he called the genes as factors. So this is quite a little bit history of Mendel. So let us now go to the experiments of Mendel. So you should also go through this brief history of Mendel before we start learning about heredity or genetics. Who introduced it or who started this genetics, it should be known. Now, why did he select pea plant for his experiments? Now, we all know that Mendel, he selected garden pea plant for his experiments. So, what are the characteristics of a garden pea plant for which he has selected pea plants? So, pea plants have several distinct varieties, and they all have a sharp contrasting characters such as their color, we can say their shape of the seeds. They are contrasting. Pea plants always bears bisexual flowers. That is, each flower is having both male and female parts. So, pea plant is bearing bisexual flowers. Each flower is having male and female parts. The structure of the flower is such that this completely encloses the reproductive organs until fertilization. So, which helps in self-pollination. Now, the next character is it is easy to get the pure lines for several generations. And during study, each pea plant grows over a short span of time. So, it is easy to note the results one generation after another in a short span of time. Flowers of pea plants are adequate in size and they are easy to handle. Each plant can produce a large number of seeds. So now these are the 7 main important points on which based on which Mendel has chosen pea plant for his experiments. So please go through it and make it clear that what are the 7 main characters for which he has chosen the pea plant or the garden pea for his experiments. So let us see the main contrasting characters what he has taken. Now here we start with the 7 pairs of contrasting characters in pea plant studied by Mendel. So we have taken 2 types of expressions, that is, one is dominant and one is recessive. First we will deal with the flower color. So one dominant was purple color, recessive was the white color. Here plant height was tall, dominant, and recessive was short. Seed color, dominant is yellow and recessive is green. Next comes the seed shape, dominant is round and recessive is wrinkled. Again comes the pod color, dominant is green and recessive is yellow. Next comes the pod shape, dominant is inflated, full, and recessive is constricted or wrinkled. Now dominant flower position is the axial position and the recessive is the terminal position. So these are all 7 pairs of contrasting characters which Mendel took for his experiments. Now in this video, it's already a very big video for the introduction of heredity. Now we will move on to the experiments in the next video. So till then, you get all these topics and all these definitions and points clear in your mind, so that when we start on with the experiments, different experiments, they like monohybrid and dihybrid cross, then it should not be a problem for you. So you should be clear with all these characters, you should be clear with all the terms of heredity, and you should be clear with the basic concept of all the main topics which I taught today. So please go through all of them, and don't forget to subscribe to my channel so that you get the updates whenever I post it. So please do like and share, and don't forget to share your comments also. Thank you.